But let's get started. So first of all, we're going to be talking about anatomy and diversity, specifically in primates. Some key terms, we've talked a little bit about these before, but just to kind of reiterate, strepsirini, these are going to be lemurs and lorises and other monkeys that have moist noses. Haplorines are going to be tarsiers and simians. They're going to have dry noses. Within haplorines, we have platyrrhines. These are going to have flat noses with sideways facing nostrils. These will be tamarins, spider monkeys, capuchin monkeys, cebidae, and squirrel monkeys. And then catarrhines, these are going to have hooked noses with nostrils facing downwards. So your old world monkeys, the great apes, baboons, the greynon, macaques, and even humans. Then cercopithecoids, this is a family of mammals, that means old world monkeys. Hominoids, which is a primate group that includes humans, our fossil ancestors, and the anthropoid apes. And then hominin, which is part of the family or the larger group of primates called hominids. Now, hominids include orangutans, gorillas, chimps, and human beings. However, hominins really only include specific human ancestors and then humans. It's kind of the square and rectangle thing that you learn in math. All hominins are hominids, but very few hominids are hominins. Here you can see a little bit more about the differences between strepsirini versus haplorini. And then this little cladogram here kind of shows you how everything branched off. Now we're going to go over some key terms about dentition. Most of these aren't super important for you guys to know, but they are leading up to something, I promise. So to start off, we have enamel. This is that kind of hard, glossy substance that covers the crown of the tooth. This is what we try to protect by brushing our teeth. Then we have cementum, and this is a very thin layer of bony material that helps fix your teeth into your jaw. Now, a lot of times when forensic anthropologists get human remains, they are oftentimes missing teeth. And this is because whenever you die, your cementum actually erodes away. It does this even quicker if remains have been found outside where they have been exposed to different weathering conditions. Without that cementum, your teeth get very loose. It's very easy for them to fall out over time. Then we have dentine. This is a very hard, dense, bony tissue that forms the bulk of your tooth and sits beneath the enamel. Crowns are the part of the tooth that actually projects from the gum, so it's the part that you are actually brushing that you can see. The neck is a narrowed part of the tooth between the crown and the root, so it constricts just a little bit. The root is the lower 2/3 of a tooth, and they're normally buried in bone, so either in your maxillary bone or your mandibular bone, and they help to kind of anchor your teeth into their position. The occlusal surface is the portion of your tooth that comes into contact with another tooth in the other jaw. So it's basically just that top of your teeth that you use whenever you bite or chew something. Cusp. These are these little bumps that you see in teeth. It's an occlusal or incisal eminence on a tooth. Canine teeth, sometimes known as cuspids, each possess a single cusp, whereas premolars are sometimes known as bicuspids because they typically possess two cusps. Now, molars often possess either 4 or 5 cusps. Sometimes you will see molars that have more cusp. This is sometimes due to medical issues, as well as just other genetic factors. Occasionally, you will see molars that have less than 4 cusps, but that's a little bit more rare. But the average human, quote unquote, has 4 or 5 cusps. And then the crest is the maxillary ridge that passes along the alveolar process of the fetal maxillary bones. Basically, it's that kind of bumpy part on the top or roof of your mouth. If you feel it with your tongue, you'll feel where there's kind of like a line. And that actually fuses as you get older. Now, what we're building up to is talking about dentition formula. So you can actually count the dental formula for any animal. Humans have a 2, 1, 2, 3 dental formula. And the way that you count these is that the first number is counting the number of incisors. The second is your canines. Third is your premolars. And then finally, the fourth is your molars. Now you might want to argue that humans actually have 4 incisors and 2 canines and 4 premolars and 6 molars. But that would be a lot. So we actually just count half of the teeth. So you literally split it in half and count just one side of the jaw or the maxillary surface. Like I said, humans have a 2, 1, 2, 3 dental formula. That third molar is actually your wisdom tooth. Now, it used to be that everybody had wisdom teeth pretty much, and it was because we were eating much harder foods that wore our teeth down very quickly. And so you would get a wisdom tooth later on in your life than you would the rest of your adult teeth. That would kind of help replace the molars that had worn down so that you could still continue to eat. We also had wider jaws back then. However, as humans have started to eat a much more processed and soft food diet, our jaws have gotten smaller. Because of this, we don't have as much room for third molars, but we also don't wear down our other molars as quickly or as severely as our ancestors did. So that third molar isn't really necessary anymore. It's actually a very interesting case of evolution still occurring as well, because humans are actually evolving to have wisdom teeth less often than we used to. So every year we're seeing people born that never develop wisdom teeth at all. Now, the fact that our jaws have gotten smaller have also meant that we don't have as much room for wisdom teeth, which is why if you do have them, people typically get them removed because they crowd and we just don't have the space for them, so they cause other issues in our mouths. Now, not all primates have this 2, 1, 2, 3 dental formula, and not all primates have a dental formula that matches between the top and the bottom teeth. You will see this on the homework for this week as well. But we will go over it very quickly together. So when you're trying to decide between if something is a non-primate or a primate, you can tell if they are or not, because all primates have a bony rim that completely encircles their orbits. This is the eye orbital. All primates also have forward-facing orbits. So we have forward-facing eyes. And then we have a relatively large brain compared to non-primates. Now, as we get more specific, the differences between Strepsirrhenes and Haplorhines. Strepsirrhenes tend to have longer snouts and a greater reliance on smell. They often have tooth comb incisors. These are just very long, thin, closely set together incisors that are actually used to kind of comb through fur, comb through leaves to catch insects. So it's also a good way of telling if something is an insectivore or not. If it has a tooth comb, it is likely an insectivore. Strepsirrhenes also have an ectotympanic ring, while haplorhines have an endotympanic ring. Strepsirrhines are often nocturnal, whereas haplorhines are usually diurnal, so they are awake partially during the day and partially at night. Haplorhines also have a fused mandible, as well as an enclosed eye socket. So this is that enclosed eye orbital. This is why in humans we actually have where our eye sockets sit, we don't have an opening at the back. It completely encloses our eyes. All haplorhines have this. A lot of strepsirrhines don't. This is also sometimes known as a post-orbital plate. Now, between tarsiers, platyrrhines, and catarrhines, catarrhines have downward pointing noses, whereas platyrrhines have outward pointing noses. Platyrrhines also have prehensile tails, whereas catarrhines, if they do have a tail, it isn't prehensile, it tends to be shorter as well. Or more often, they just don't have a traditional tail at all. Catarrhines have 2 premolars and that 2-1-2-3 dental formula, just like humans. Platyrrhines have 3 premolars and a 2-1-3-3 dental formula. Now, tarsiers, I will mention again, they get a little bit weird. So they actually have a 2-1-3-3 dental formula on the top, or their maxillary dental formula, and then a 1-1-3-3 mandibular dental formula, or bottom. They also have very large eyes relative to their brain size. Incredibly large eyes is indicative that something is nocturnal, as well as that it is an insectivore. Now, between cercopithecoids and hominoids, hominoids tend to have relatively larger brains than cercopithecoids, as well as having 5 cusps on their lower molars and a Y5 fissure pattern. Now, you can look this up, but essentially it just means that in the middle of a molar, there is kind of a Y shape that you can see, and it's what helps divide that molar up into 5 cusps. By the way, humans typically have a Y5 fissure pattern, but there are actually a couple different fissure patterns that humans typically have. Y5 is just one of the most common. Now, between hominoids and hominins, hominoids tend to have lower third premolars that are single cusped. They also have lower faces that project more than hominins, so they have more facial prognathism. They also tend to have larger canines that are honing, so it is more of an indicator of sexual dimorphism that hominins don't typically show as much. Hominoids also have relatively smaller brains compared to hominins. Once we get to hominins or early human ancestor, this is when we start to see brain capacities really shoot up, which makes sense because this is also when we start to see more refined tool making, more refined fire use and cooking of food, as well as structure making and social groupings. And then hominoids have a foramen magnum that's placed much further underneath the brain case. The foramen magnum is actually this little hole at the bottom of your skull that is where your spinal cord kind of helps attach your head to your body. Now, as something becomes more bipedal, we actually see that foramen magnum move to be more centrally located underneath the skull rather than further back, which we'll talk about in the next lecture as well. Now, when you do the homework, or Lab 9, I should say, you guys will need to go to this eSkeletons website. It is also linked on the additional resources folder on Blackboard. You will be looking at their skulls and their dentition, and then trying to determine if they are primates or not, whether they belong in the Strepsirrhini or Haplorhini group, then whether they are in the Platyrrhines or Catarrhines group or tarsiers, the Cercopithecoids or the hominoids, and then if they are hominins or not, and why. So you have to give a couple reasons, a couple traits that helped you place them into each category.